We're going to analyse our protein by Sds Page. This is polyacrylamide gel electrophoresis. So first need to check we've got the right kind of gel. So this has a 4 to 20% acrylamide gradient. It's got 10 well formers and each well will hold 30 microliters. So we open up the packet and get out the gel. And first thing to do is just dry down the outside of that gel cassette because it's quite slippery because of the SDS. And then we can start to pull out the well formers or the comb. This is a bit tricky to do. Just try and be as gentle as you can so that you don't disrupt the wells, which are fairly fragile. Next step, very important, is to peel off the green sticker on the bottom. So otherwise you won't get current running through the gel. Okay, we next get our electrode assembly, which is shown here. Note that it already has a buffer dam attached to one side. So this is like a blank gel cassette that's going to hold the buffer inside that electrode assembly. Also really important to put the gel in the correct way around. There's a right way and a wrong way. So the wells should be facing inwards. So the low side of the gel cassette on the inside. And we can check this is correct by looking at the numbers for the wells, and they should be the right way round. We next put the electrode into the tank, and it should be on the side towards you, and you should have the red terminal next to the red on the gel tank. So we're going to pour the buffer in now. We tilt the tank to do this. This is to minimize bubbles. So the buffer has SDS, which is a detergent. We don't want it to make excessive bubbles. These can potentially interfere with the current flowing if they get under the actual electrode. So we fill out the outside tank to about a third of the way up, and then we fill up the inside of that electrode assembly. And this should come all the way to the top so that it covers the wells. So this is known as a discontinuous buffer system. There's a separate outside and inside, even though both of them get the same buffer. Okay, next we need to prepare the samples. So just like in an agarose gel, we need to mix them up with some loading dye in order to get them to load properly into the gel. So here we've got some cell extracts and some PFU protein. We label our tubes before starting, just like always. And we're going to mix 10 microliters of loading buffer with 40 microliters of sample. So this loading buffer contains a density agent and a dye for tracking, so in that way it's like the buffer we use for agarose gels, but it also contains SDS and some other ingredients which help to denature the proteins so that they run according to their molecular weight and not according to their shape. So we just mix up our buffer with our samples here in a second set of EpiTubes. Just note how she's moving the tubes along one at a time in the rack so she can keep track of where she's up to. Okay, then these go off to be heat treated. And the heat treatment in combination with the SDS denatures the proteins, and after they've been denatured they're ready to load on the Sds Page gel. So we're just going to make a plan of where we're going to load our samples. So know that there's bench coat on the bench here, so this paper and plastic covering, so it's fine to write on the bench coat. Please don't write on the bench itself though. You could also write this into your lab archives or onto a disposable plastic sheet or something like that, or a reusable plastic sheet. Okay, so we're going to load some cell extracts, 5 microliters of those, 20 microliters of our purified protein, and very important, we need to load a marker as well. In this case, the marker is a standard set of proteins that have known sizes. So we're going to pipette very carefully into the top of the gel. So note that this gel is a vertical gel. So we load it at the top and then it runs from the top to the bottom, unlike an agarose gel where it kind of runs from side to side. So here's a close-up view of the wells. You can see the loading. So again, the pipette tip is just at the top of the well. You don't need to force it all the way down. The density agent in that loading buffer will help the sample just fall into the well and try and minimize the amount of carryover into the neighboring wells. So once we've loaded all of our samples, we're going to now run the gel. So we just plug it into the power pack, put the lid on first obviously, plug it into the power pack, set the voltage, and away it goes. And just like with agarose gels, we check for bubbles as a good indicator that we've genuinely got current flowing. And again, this just shows the electrolysis of the water into hydrogen and oxygen. So here, this is the gel running in time lapse. You can see the bands of marker dye running down from top to bottom. And when it gets close to the bottom of the gel, we're going to switch it off and then we can go and image the gel.